has acquired resistance to a particular antibiotic, it passes the resistance allele to subsequent daughter cells that result from binary fission. In addition, bacteria that have acquired a trait such as antibiotic resistance can transfer this allele to other bacteria through any one of the three mechanisms: transformation, conjugation, transduction. So transformation, when bacterial cells die, they frequently lyse, that is burst, releasing the intracellular contents, including fragments of DNA to the environment. These fragments can be taken up and incorporated into the chromosome of a living bacteria to provide the recipient with new characteristics. This process is called bacterial transformation, and if the incorporated DNA contains genes that encode for resistance to an antibiotic, a previously susceptible bacterium can be transformed to now be resistant. The video of below 23 seconds provide a quick overview of transformation. Conjugation. Many bacteria have plasmids, which have smaller pieces of DNA separate from the primary bacterial chromosome. These plasmids can carry genes that provide resistance to antibiotics, and bacteria that contain plasmids are able to conjugate with other bacteria and pass or replicate to recipient bacteria. The electron micrograph below shows two bacteria that are joined by a temporary hollow tube-like connection called a pilus. Here is the animation illustration which of a bacterial conjugation. Let's watch.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
Now we go to the next portion. In 1968, a Shigella epidemic killed 12,500 people in Guatemala. The Shigella bacteria that caused the outbreak had a plasmid carrying resistance to 4 antibiotics. So if we need details on that, we could go by the CDC webpage on Shigella. Let me see if it's of use to us. We can use it, or else we come back. Let's try to link. Yeah, we have connected to the Center for Disease Control, CDC, on Shigella. So this is Shigellosis, Shigella infection. Okay, this is the Shigella, the actual picture of the bacteria. These are bacteria germs that cause diarrhea. The most common ways people get sick are from eating or drinking contaminated food or water in contact with someone who is sick or has recently been sick with Shigella infection. Shigella can also spread during sexual activity with a sick person, right? So just a general mention there. And now we go back to the subject matter proper. And we go to the next subject called transduction. Genetic information can also be carried from one bacterium to another virus. Bacteriophages, or simply phages, are small viruses that infect bacteria and use the cellular components to make bacteriophage replicate. During the infection and replication, it is possible for bacterial genes to get incorporated into the viral genome. One of them, viral replicates carrying the bacterial allele, may then subsequently infect another bacterium and pass the new allele on. The illustration below begins with the donor bacterium that has a gene A+, which encodes for resistance for a particular antibiotic. In the second and third scenes, the viral proteins and genetic material are replicated and then self-assemble into new viral particles. In the process, however, the bacterial gene for A+ gets incorporated into one of the virus particles. The viral particles are then released from the donor bacterium, and the virus with A+ gene infects a recipient bacterium, after which the A+ gene can become incorporated into the genome of the recipient, which has now acquired this gene for antibiotic resistance. Please take screenshots. Next topic is slowing the phenomenon of antibiotic resistance. The development and spread of bacterial resistance to antibiotics is inevitable, but it could be greatly curtailed through relatively simple measures. These include preventing infection, general infection control, hand washing among healthcare workers and food handlers and the general public, modern sanitation, effective systems for dealing with sewage and providing clean water, proper food practices, and rapid identification and isolation of new cases of infection, example new cases of TB. This is particularly important with drug-resistant cases. Continued development of new antibiotics, decreased agricultural use of antibiotics to enhance growth, physician education to reduce inappropriate prescriptions and inappropriate use of a broad spectrum of antibiotics, educating physicians and patients about the importance of taking the appropriate dose of an antibiotic for the full period of treatment that is indicated, consumer education regarding the importance of bacterial resistance and the uselessness of taking antibiotics for viral infections such as the common cold. Drug resistance and HIV. As noted earlier, HIV is a retrovirus consisting of a single strand of RNA inside a protein coat. When HIV enters a CD4 lymphocyte, it sheds its protein coat and uses a viral enzyme called reverse transcriptase to create a segment of DNA using the viral RNA as a template. This double-stranded DNA version of HIV then gets incorporated into the DNA of the infected host cell, and process is called reverse transcription. Let me read that again for you. This double-stranded DNA version of HIV then gets incorporated into the DNA of the infected host cell, and this process is called reverse transcription. This has important consequences for the development of drug resistance by HIV because of several key characteristics of HIV. Right? Going back to the point there, as noted earlier, HIV is a retrovirus consisting of single strand of RNA inside a protein coat. So when HIV enters a CD4 lymphocyte, it sheds its protein coat and uses a viral enzyme called reverse transcriptase to create a segment of DNA using the viral RNA as a template. Remember this. So this double-stranded DNA version of HIV gets incorporated into the DNA of the infected host cell. This process is called reverse transcription. This has important consequences for the development of drug resistance by HIV because of several key characteristics of HIV. HIV replicates at a prodigious rate, producing billions of new virus particles each day. Reverse transcription is notoriously error-prone, leading to frequent mutations. Current antiviral treatment regimens control the infection, but they do not completely eliminate the virus. Given the persistence of HIV, the high rates of replication and high error rates during reverse transcription, mutations in HIV are inevitable, and some of these mutations lead to the eventual development of drug resistance. HIV infects CD4 cell lymphocytes and hijacks the cell's machinery to create viral proteins and RNA. HIV's genetic material in RNA, and in order replicate reverse transcriptase results in very rapid production of new HIV particles. Two concepts are important to understanding of the development of drug resistance. First, HIV infection is characterized by high levels of virus production and turnover. In most untreated patients, the total number of productively infected cells in their lymphoid tissue has been estimated to be approximately 107 to 108 cells. During the chronic phase of HIV infection, this number is relatively stable, reflecting the the balance between the infection of new target cells and their clearance. Because the half-life of infected cells is remarkably short, 1 or 2 days, the maintenance of this steady state requires that HIV-infected new target cells at a very high rate. Second, the viral population in an infected person is highly heterogeneous. The reverse transcription of viral RNA into DNA is notoriously prone to error, introducing on average 1 mutation for each viral genome transcribed. Most of these errors are base substitutions, but duplications, insertions, and recombination can also occur. The high rate of HIV infection combined with the high mutation rate that occurs during each cycle of infection ensures that patients have a complex and diverse mixture of viral quasi-species, each differing by 1 or more mutations. Under these circumstances, it is easy to understand why, if any of these mutations can confer some selective advantage to the virus, such as a decrease in its susceptibility to an antiretroviral agent, the corresponding quasi-species will overtake the others following a simple Darwinian selection process. The next subject of interest and importance would be control of gene expressions in transcription and translation. And before we could proceed, let's just watch a short video, right? So you get a general idea what transcription and gene expression is all about. And then we proceed on to the subject matter proper, right? Okay. Let's just watch it for a short while.
In order to properly function and survive, living things must express their genes to make protein. Some genes need to constantly express their proteins, but other proteins only need to be made some of the time. Prokaryotes and eukaryotes have different ways of regulating their gene expression. Prokaryotes rely on variations in their environment to regulate their gene expression. For example, if lactose is present in the environment, it will trigger the expression of lactase so the bacteria can break down lactose. If lactose is absent, the gene is not expressed. Eukaryotes also regulate genes in response to their environment. Each cell of a eukaryote only expresses a small number of genes in the nucleus that pertain to its job. Transcription factors are proteins that regulate gene expression by binding to specific sequences that are associated with the transcription initiation site. Transcription factors can be activators or repressors. Along the DNA strand, there are regions of DNA that are called enhancers and regions called silencers. Enhancers are regions of DNA that increase the rate of transcription when an activator binds to them. Silencers are regions of DNA that decrease the rate of transcription when a repressor binds to them. The environment of the cell, and even of the whole organism, can affect gene expression. The Siamese cat is a perfect example. Siamese cats have a mutated allele that codes for the enzyme tyrosinase, the first step in the production of fur pigment, and it's affected by temperature. When the temperature drops, gene expression increases, and the pigment in the fur also increases. When the temperature increases, gene expression is reduced, and the pigment in the hairs decrease. Nucleosomes also play a role in gene expression. When the tails of the histones are acetylated, the nucleosomes become less tightly packed. In this loosened form, transcription can occur, which is the first part of gene expression. When the tails of the histones are deacetylated, the nucleosomes pack in tighter, stopping gene expression. Additionally, methylated cytosine will tighten the nucleosomes, while unmethylated cytosine will loosen them. So, active open chromatin and unmethylated cytosine, along with acetylated histones, will activate a gene and turn it on. Silent condensed chromatin and methylated cytosine, along with deacetylated histones, inactivate a gene and turn it off. Genes are the sequences of DNA which transcribe into RNA, typically becoming protein during translation. Genes have three main parts: the promoter, which is the transcription initiation site, the coding sequence, which is transcribed, and the terminator is the site where transcription ends. DNA is double-stranded, but only one strand is transcribed for protein synthesis. The coding strand is called the antisense strand, and the non-coding strand is the sense strand. Transcription begins at the promoter on the antisense strand. The RNA forms in the 5' to 3' direction, reading the antisense strand in the 3' to 5' direction. RNA polymerase binds to the promoter, unzips DNA by breaking the hydrogen bonds, and covalently joins the RNA nucleotides together. Base pairing between DNA and RNA is still between C and G, A and T, but on the mRNA strand, uracil replaces thymine. When messenger RNA is formed in prokaryotes, it's immediately ready for transcription. But in eukaryotes, the mRNA is referred to as pre-mRNA and needs to be mRNA. The sections that are removed are called intervening sequences, or introns, and the sections that are kept are called exons. After the introns are removed and the exons are spliced, the mRNA is mature and ready for translation into a protein. Eukaryotes can selectively remove exons of a gene to form different proteins from the same gene. This is called alternative splicing. This greatly increases the variety of proteins that can be produced. Strangely, only 1% of DNA actually codes for protein. The rest of the DNA is called non-coding DNA and used to even be called junk DNA in the past. But just because DNA doesn't code for protein doesn't mean it has no purpose. Here are 5 different uses for non-coding.
Deacetylate DNA with telomeres, introns, non-coding RNA genes, and gene regulatory sequences. Satellite DNA include tandem repeats, which are the portions. Used for DNA profiling, telomeres are repetitive DNA at the end of chromosomes that protect the chromosomes from deterioration during replication. Introns are non-coding sequences. Within genes, non-coding RNA genes can form structural RNA like the tRNA molecules, and gene regulatory sequences includes regions of the DNA that are used in the process of transcription, such as promoters, enhancers, and silencers. Thanks for watching this episode. Right now, proceeding with the subject matter proper, transcription and translation are the two main steps of gene expression, which is the process by which cells read and express genetic instructions in their genes. Transcription, the enzyme RNA polymerase uses DNA as a template to produce a pre-mRNA transcript. This transcript is then processed into a mature mRNA molecule. Translation, the mRNA is read by a ribosome and which translates the mRNA sequence into a chain of amino acids that form a protein. There are some things to know about transcription and translation. Transcription controls gene status. Transcription is a key step that controls when genes are on or off, which in turn determines the identity and status of the cell. The transcription translations are sequential. Transcription and translation are carried out in consecutive stages, which makes it difficult to distinguish between them. Transcription and translation are complex. Both processes involve multiple protein complexes that are recruited or replaced as the process progresses. Gene expression can be regulated by controlling RNA production, and the gene expressions vary by organisms between prokaryotic and eukaryotic categories. Prokaryotes are single-cell organisms like bacteria, while eukaryotes include multicellular and unicellular organisms like animals, fungi, plants, and protists. It consists basically this consists of two major steps: transcription and translation, where the gene expression. Together, transcription and translation are known as the gene expression. During the process of transcription, the information stored in a gene's DNA is passed to a similar molecule called RNA, ribonucleic acid, in the cell nucleus. Now, what is gene expression? Transcription and translation. The central dogma of gene expression includes two sequential steps: transcription DNA to RNA and translation RNA to protein. Transcription is the key step that controls the on and off of genes and subsequently underlines the identity and status of the cell. Right? Now we're going into the subject matter proper through the PubMed Central, the international research portal. Right? Now, control transcription, control gene expression. Right? The transcription is the epicenter of gene expression. The complexity of a living organism is not driven by gene number, but gene regulation. Controlling which genes to express and to what extent dictates the subsequent cell identity. Transcription of the cell critical initial stage in gene expression is regulated delicately to maintain the cell status. Recent developments in the genomic approaches provides unparalleled coverage of the study of transcription. Still, basic molecular biology and biochemistry providing mechanistic insights into how the regulation is achieved. In this feature, regulation of transcription mechanisms and biological functions, the latest advance such in genetics, mRNA processing, RNA quality control, and human immunodeficiency, virus HIV, transactivation are discussed here. Now, control transcription, control gene expression. Well, a gene had been traditionally viewed as the basic molecular unit of heredity by Crick and Watson Crick, 1958 and 1970, in the form of DNA or RNA. It carries the raw genetic information that can be turned into functional products, usually proteins. However, the number of genes does not reflect the complexity of the organism. For example, a human has about 20,000 protein coding genes, which is about 6,000 more than a fruit fly, 2,000 more than Caenorhabditis elegans, and 14,000 more than budding yeast, but 10,000 less than a lab mouse, 5,000 less than the model plant Arabidopsis, and 17,000 less than rice. Clearly, the level of complexity of the organism is achieved by regulating available genes, not simply by introducing more genes. Well, the central dogma of gene expression includes two sequential steps: transcription, DNA to RNA, and translation, RNA to protein. Transcription is the key step that controls the on and off of genes and subsequently underlines the identity and status of the cell. Well, all courtesy and thanks to PubMed Central, because this much of research material is available for all of you to listen to, read, and study. All right? So please make the best use of it and go for sit your qualifiers and come out with flying colors. Now, the central dogma of gene expression includes two sequential steps: transcription, DNA to RNA, and sequential steps: transcription, DNA to RNA, and translation, RNA to protein. Transcription is a key step that controls the on and off of genes and subsequently underlines the identity and the status of the cell. Right? So I'm just reading out just the important points. Just make a note of those. That's all you need. So the total length of DNA from a human cell is about 3 meters. In a living cell, DNA is packaged into a highly compact form called chromatin. Genes are not active when they are compacted, when they're in compacted form. So the factors involved in opening and closing specific regions control the accessibility of genes. A set of factors called polycomb group (PCG) protein is involved in modifying the chromatin structure and subsequently regulating a large number of genes. The dynamic nature of multi-subunit protein complex and its complex functional impact is described in detail here. While gene regulation does not stop at finding the gene, the transcription machinery finds an accessible promoter and assembles multi-megadalton protein complex to initiate transcription. As soon as the nascent transcript emerges from the body of the RNA polymerase II, Pol2, the enzyme that transcribes protein coding genes, the RNA is protected by the addition of a 5' cap, and the descriptions show a unanimous phenomenon that takes place on every molecule of RNA transcripts. Now, formally, removal of the cap and subsequent RNA degradation are thought to only take place after the RNA is exported to the cytoplasm and translation has been initiated. This discovery has changed the way the mRNA capping and decapping are viewed in time and space. RNA degradation, or turnover, has a significant place in gene expression. Huxley and Tollerby in 2009 compared to DNA, RNA is a very short-lived species in cells. Its relatively rapid turnover provides a flexibility for organisms to react to the environment and adapt quickly to a change in the gene expression profile. Historically, mRNA degradation is only thought to occur once at least a round of translation has taken place. Now, Liu et al. Provide an updated review of the many RNA degradation pathways taking place in the nucleus. Degrading nascent RNA transcripts in the nucleus has been reviewed as a quality control mechanism to quickly get rid of undesired RNAs before they export it to cytoplasm and waste the effort of ribosomes in translating wrong patients. The start of transcription relies on promoters. The termination signal typically relies on sequence information at the end of the genes. Doing the current understanding of the sequence requirements and the protein factors involved in this process called mRNA 3' processing. It cuts the RNA of the moving Pol2 and adds a stretch of adenines to the RNA. The sequence driving this process is called polyadenylation signal or polyase signal. It's been known that the promiscuous nature of the polyase signal sequences leads to the selective use of multi-polyase signals. This feature, described as alternation polyadenylation, has been linked with many aspects of the gene expression: premature termination, RNA splicing, RNA stability, and expression level. Courtesy, Mayer and Bartel, 2009. However, the exact mechanisms that dictates alternative polyase signal usage is unclear. This article also provides a simple yet broadly applicable model to characterize how the choice among many polyase signals is made. The process of making RNA nucleotide by nucleotide transcription elongation is tightly controlled and interlaced with all the above events, and the elongation control is a critical step in determining transcriptional output instead of looking for an open promoter to start prescription. To start, transcription. That's a correction there. Upon activation, positive transcription along factor PTEFP triggers the fast-paced productive elongation that leads to mRNA production, this is therefore seen as PTEFP is therefore seen as the key for rapid amplification of gene expression. Okay, right. Transcription factors and combination control. Transcription factors, TFs, are regulatory proteins whose functions is to activate or more rarely to inhibit transcription of DNA by binding to specific DNA sequences. Transcription factors have defined DNA binding domains with up to 10⁶-fold higher affinity for their target sequences than for the remainder of the DNA strand. These highly conserved sequences have been used to categorize the known TFs into various families such as the MMADs, box-containing proteins. SOX proteins and POU factors can also be classified by the three-dimensional protein structure, including basic helix, turn helix, helix loop helix, and zinc Finca proteins. These different structural motifs result in transcription factors specificity for the consensus sequences to which they bind. Sequence-specific transcription factors are considered the most important and diverse mechanisms of gene regulation in both prokaryotic and eukaryotic cells. In eukaryotes, regulation of gene expression by transcription factors is said to be combinatorial. In that it requires the coordinated interactions of multiple proteins. These differential structural motifs result in transcription factor specificity for the consensus sequence to which they bind.